Thank you, Kemi. Yeah, I guess it's almost past five, so let's start introducing Krishna and we can get us started. So yeah, I'm delighted to have Krishna here with us at the department. Yeah, so I guess like Krishna's official appointment or position is Krishna is the Honse and Vivian Lim Professor of Engineering at Stanford. He's with the departments of Electrical Engineering and by courtesy Bioengineering and Neurobiology. And he's also a Howard Hughes Medical Institute investigator. A little bit about his background. Krishna did his bachelor in UC Irvine, his PhD in MIT, and then a postdoc at Caltech before he joined Stanford's faculty in 2001, I think. And he hasn't moved from there since that time, although he's gone like all the way up to the top of the ladder. So Krishna basically focuses his research, or a lot of his research, on understanding the neural basis of movement. And his group pioneered like this kind of revolution in systems neuroscience that consisted in stop thinking about what each individual neuron does at a time and thinking more about the collective behavior of neural populations. And this was very impactful in the motor control community, and ever since has extended to what many people would call like higher functions, like making decisions or things like that. And in parallel to this, like population revolution in systems neuroscience, he also translated many of these developments to the brain-computer interface field to help paralyzed individuals, which is what he's going to tell us about today. And he's been instrumental in this translation from the science to being like advisor in companies such as Neuralink and Control Labs that we're now hearing about every day. Just to give people some measure of Krishna's scientific impact, I did a quick control search in your Google Scholar before the talk. So the word nature popped up like 17 times, 55 of them as nature papers, and many of the others were nature neuroscience papers, and there was a bunch of neuron papers. So basically all these journals that us neuroscientists like. But one interesting thing about Krishna that I want to highlight is when you meet people working with him in his lab, which I've had the luck to do many occasions in conferences in the old days, is that they always highlight how Krishna is like a really great mentor. So I'm quoting one of Krishna's trainees here when he told me he's the best example of someone who fully lives by the ideal of being an advisor, teacher, role model, and friend, which I think is one of the nicest things that anyone can get, can be told basically. But of course we are scientists, so we care about data. So I want to back up this statement with some hard facts. One is according to the Twitterverse, Krishna is very generous, also lending his Porsche to his lab when they have occasions to celebrate personally. So I guess this speaks highly as helping in mental health and happiness. And also academically, people who have worked with Krishna have basically gone and populated the neuroscience community, starting very successful labs in many universities, including Columbia University, University of Chicago, Carnegie Mellon, Emory, Michigan, I think UCLA and UCSD, Google, and I'm probably forgetting a few more, but this is the people that I could recall quickly. So, I will, now that I've told you about Krishna's influence in the field, I will let him present you his science so you can appreciate why this is the case. So Krishna, thanks again for being here. The floor is yours.
Thank you very much for the overly kind introduction, but indeed it means a great deal to me that the people I interact with know they're as special as they truly are. So I think everybody can see. Can everybody hear me okay too?
Yes.
The volume's about okay? All right, great. Okay, so thank you again so much. And this is my first time at Imperial, although I've spent many, many weeks visiting our close friend and collaborator Manish Sahani over at Gatsby and so forth, and really love London and in England, and so it's always a pleasure to be here. I wish I were there physically. Soon I will be, I'm sure. Okay, so today what I wanted to do is not try to overload with too much and try to give a survey of everything, but really focus in on two stories. And those two stories center on the human abilities of speaking and handwriting. And the simple tagline, if you will, that I'd like to leave you with, so let me tell you up front, is really rapid, highly dexterous motor sequences. Okay, so how do we go quickly through things that require a lot of coordination, like handwriting and speech? Okay, and do that in fast sequences. And this is something that we've always longed to do in our non-human primate work, and we're able to do now because it's so closely related to the prosthetics work of our clinical trials that we do this in humans. Okay, so I'll just give a brief introduction to the concept of brain-machine interfaces for those of you that very understandably may not be familiar with this, and then I'll dive into these two stories. So this picture of a famous actor from a couple decades ago, Christopher Reeve, who played Superman in the movies, was thrown from a horse, broke his upper spinal cord, neck, and from that day forward was not able to walk or move his hands. But a little less appreciated is that he was also not able to speak well because he needed to be on a ventilation system. And many people are like this, as the pie chart on the left show, regarding paralysis, and would simply put, we'd really like to help these people, okay, through neuroscience and engineering, but one day have a sight line to trying to help people that are in profound need, okay? And this picture also brings to mind this sort of presumably almost ancient idea that, hey, what goes on in your brain or in your head may still be there, even if you can't move. It's just that those signals can't make it out the spinal cord and down to the muscles. And this side schematic illustration unpacks that a little bit more. So let's say that you want to reach out with your arm and pick up a cup of coffee. Well, you know where that cup of coffee is because you can see it, and you can calculate all sorts of vectors between your hand and your eye and your object that you're wanting to reach towards. Those plans to move your arm are then brought forward to the premotor cortex and the motor cortex, where these plans are elaborated into detailed movement sequences that go down the spinal cord and contract the muscles and cause the movement. Now, of course, if there's an injury, what we'd like to do is eavesdrop in on the electrical chatter from many individual neurons. And that's illustrated here, where these action potentials are these little blips. And we typically record from 2 of these 100 electrode arrays. That's the current technology, the so-called Utah arrays that are approved for human use. But of course, that needs to advance. I'll say a little bit more of that in a second. That electrical activity, if we understand enough about how the brain encodes information, like how you want to move your hand or your arm or your fingers, we can decode it, we can flip it around. And this is fundamentally simply Bayes' rule. If we know the probability that a given movement causes a given set of action potentials, that if we're given those action potentials, we can ask, well, what are the chances that you want to move in each direction? So it's actually quite simple, okay? Now I say that, but of course there's, 10 or 15 PhDs that are written on the exact ways to do these things, okay? They can be implemented in low power electronics, circuits, chips, okay? And the key there is not that they can be implemented in this hardware. Of course they can be. We do this all the time even on our laptops or our phones. The point is that it can be done in extremely small form factor at very low power, such that it's absolutely thinkable to take this whole processing, which you might think of as, oh, that's so much effort, it could never work, but it's possible to go ahead and put that right on the backside of the electrode array and implant the whole thing, and then wirelessly send out those signals. So that's, again, sort of the engineering development site path to how you could have these types of systems fully implanted and very safe for people. Now, what do you do with those signals? What you do is, well, you can do anything that is not captured here, okay? But three things are: you can functionally electrically stimulate the muscles, meaning, as shown here by some wonderful work by our colleagues that we're not involved with, Bolu Ajiboye, Frank Willett, and Bob Kirsch at Case Western, you can record how a person wants to move and then generate sequences of electrical stimulations to deliver to implanted wires, stimulators, in the muscle bodies. And by sequencing this, it's like you're sequencing your natural arm movement, and you can reanimate the arm to pick up cups and move them to your mouth and so forth. So this is a very promising way to reanimate, if you will, the natural but paralyzed arm. Another idea is a prosthetic arm or robotic arm, and work by Lee Hochberg, our colleague, Jennifer Collinger, colleague at Pittsburgh, Hochberg is at Brown, and MGH, shows that these signals can again be picked up and used to move an arm. And this isn't a terribly realistic looking arm, but they can be, of course. Now, we don't focus on this at all. Instead, we focus on this third path, which is a computer interface, okay? So why is that? Well, 10 years ago, it was sort of hard to motivate why, but I think today, most people understand. If you are honest with yourselves about how much time you spend in front of a device, a computer, a tablet, an iPhone or a smartphone, it's it's sort of depressing, actually, right? That's what we do a lot. So the point is that people who want to return to the workforce, which is most people who want to be able to communicate efficiently with their physicians and loved ones and everything, you need fast and accurate ways to interface with computers. Now, to orient some of these techniques, I'm showing you an axis that we can use to start thinking about performance. Because just showing that these systems can work in a lab, we and others have done that for the last 20 years. But it's really about how well can they work? How long can they work? How robustly do they work? That matters to a person. And this is words per minute that you can communicate from not at all through normal conversational presentation style speech, which is sitting above 100 words per minute. And a sip and puff interface is, as you might imagine, it means simply a straw that a person with paralysis can inhale or exhale from. A pressure sensor detects that one bit of information, whether it's in or out, and can be used for things. And that can be used to communicate at a low rate. Some of our own work to use a two-dimensional cursor on the screen and slide it around onto letters on an on-screen keyboard and then hit click, was able to get up to about 8 words per minute. And in a story that I'll share with you today, the second one, the handwriting task, this is now up around 17 or 18 words per minute. But we have a long way to go. Look at this, okay? And so maybe speech is a great way to go, or 10-finger typing is a way to go. So we need to fill this in and give people a variety of options. Okay, so this is my way of saying things are working better than many would have thought, I think, but we have a long ways to go. All right, final slide before we shift into the first story is recording technology. And I show this because this question very understandably always comes up, which is, well, Krishna, if you could measure from enough neurons, I believe you, and the problem should be simpler, but how many neurons can you measure from, and what are the prospects for new and different technologies, okay? And in the upper left here, Juan, you're able to see my cursor, are you? Yeah, perfect. Okay. So this is the so-called Utah electrode array, which many groups around the world use in non-human primates, but also people, because they're so-called 510K approved, which is a US FDA designation, saying that you can implant them, with huge appropriate surgical apparatus and so forth in people. Okay, so you can see that in 4x4 millimeters, you have 100, a 10 by 10 grid, in other words, array of about 1.5 millimeter long electrodes. And at the tip of each electrode, you can typically record from one neuron, sometimes 0, sometimes 2. And at the beginning, like in the first few months, you can get really good single units that you can declare are single neurons. After that, it's hard to spike sort, meaning tell different neurons apart, but you're absolutely measuring from action potentials. And as I'll share with you later, we routinely do this in people and in monkeys for years, okay? So these sensors don't just like spontaneously disintegrate and don't work. It's just that they don't last decades, and they don't record from thousands of neurons as you would want to pull out the most information. Now, another technology are so-called neuropixels, okay? And many of you may have heard of these. These have really revolutionized the mouse neuroscience world. These are made by IMEC, Brun Dutta, and HHMI. And what Mike Shadlin, Taryn Moore, Doris Chow, and I did as part of Hughes is throw a bunch of money into it to redesign these for monkey use, meaning they're stiffer, they're longer, there's more channels. It'll be a great workhorse for recording from the non-human primate brain, which is just much larger, okay? And so that's the 3-year project. They're now done. They work well. We're finishing qualifying them, and then they'll be available to the whole world to buy, okay? Not for profit, right? So just at cost. And what these do is that they put so many electrodes so close together, so each black square here is about 20 microns from the next, that a single neuron firing a single action potential can be picked up on multiple different electrodes. So each row is the voltage trace of an electrode. And you can see that neuron event from multiple electrodes, and that gives you an exquisite ability to say that it's that neuron and no other neurons. In fact, it can tell you with such high confidence what the exact shape of it is. They're able to not only say if it's a putative parameter or interneuron, but about 7 different classes of neurons, okay? So all these papers are coming out. It's a great way to also look at structure, because what you can do is you can do a current source density analysis. You can then correspond it to superficial, deep, and white matter, probably getting close to which of the 6 layers of cortex it is. And you can make simple observations that just lay to rest age-old questions. So for example, there's an incorrect, in our opinion, assumption out there that M1 is like V1. So in primary visual cortex, if you record surface normal down a so-called column, this is a classic, right? This is a Hubel and Wiesel absolute classic, a cortical column. All neurons along that track will have the same tuning direction. They'll all respond to rightward movement, but another column will respond to leftward movement. And there's actually not so much debate, oddly enough, but it's been believed that M1 is columnarily organized too. This is what Apostolos Georgopoulos, for example, has said. But if you just stick one of these in, and then you plot in the color code the direction that each neuron represented by each circle has its maximum response for, and the modulation depth, how much it responds, is the color circle, you see just a complete mishmash of colors. Okay, now of course this is all borne out in detailed statistics across multiple animals and hundreds of recordings and on and on and on. But columnar structure is not there. And this is actually awesome news, because you stick one of these in, you get sampling from all directions. You can do population neuroscience, and prosthetics with just one of these. Okay. Now, finally, and with full disclosure, as Juan was saying, you're now starting to hear about a couple of companies, a little bit more than years ago when we got started with this. So I'm technically a co-founder, just for full disclosure, but on a daily basis, I'm an advisor to Neuralink. And their proposition is to say, okay, what we really need is to invest heavily on being able to get up to thousands of neurons being simultaneously recorded. We need to be able to surgically, robotically implant those to use computer vision and optical coherence tomography to avoid vessels, okay? And then have electronics that are so low powered as well as wireless that they can all be implanted under the skin so you can't see them. So this is from a recent, it's a company, so it's not a paper, it's just a demo, right? But this is true stuff. I know the data very well. Okay, and what you're seeing here is just rotating around 64 of these columns, because there's 64 of these so-called threads, where a needle pokes a thread in and the needle pulls out, leaving behind this totally flexible thread. And there's 16 electrodes up the side. So 16 times 64 is 1,024 electrodes. And then if you rotate, so that's a three-dimensional sampling. And if you rotate up on the top, you can tell where in the cortex each one of those electrodes went in, okay? So if this were done in our lab, well, first of all, we couldn't, but if it were done in our lab, this would be a laborious picture to put together. Meanwhile, if you do all the scale up and automation at an industrial scale, you just get these things out, they just pop out, okay? Because you have to track everything. And this is just a demonstration of having this electrode array. In fact, there's another one on the other hemisphere that we haven't even talked about. And the electronics for recording and then transmitting all the action potentials that come out is implanted in this animal about 2 months prior. And you can see that he can move a joystick to play this game, okay? And in fact, he can do that neurally only, okay? And here you can see him not moving any joystick. It's neurally only, and he's playing this right-sided Pong. This is not sped up. This is not hard to do. And the reason that you can get very high performance, including things like this Pong game or grids and get high bit rates, is because you have such vast information up front, okay? Now, why do I show all this? I want to make sure to leave you with the impression, which is true in my opinion, that there's a huge amount of technology that's just about to be unleashed on the community in an exciting way that will enable all of us to take the science to the next level, okay? So what about a handwriting BCI? All right, so we can simply ask people to attempt to move their hands. And that's exactly what Frank Willett here, who's the brilliant postdoc behind this line of work in our lab. And we all know how to handwrite. So that takes about 5 seconds to explain, okay? And what we ask our participants to do is just imagine printing like A, B, C. So not cursive or not something too elaborate, but just use your natural handwriting and write letters, okay? And we do not belabor that those letters need to be written at the same speed. That's on us. Algorithmically, we need to deal with that if there's varying speeds. And we also don't stress to have the letters written at the same size. Now, we encourage them to imagine like writing on a lined legal pad. Okay, so there's some regularity, but sometimes letters are bigger or smaller. So that scale invariance and that temporal invariance that I'm describing, we have to handle algorithmically, okay? And the reason for this is you do not want to build systems where people have to change their natural behaviors. That's been a consistent way to fail in industry. For those of you old enough to remember the Palm Pilot, okay, which was like the original electronic organizer, you could write with a stylus and it could recognize your handwriting, which is awesome. But they said, listen, if you want to do better, and you did need to do better, please adopt this different alphabet, okay, or slightly change everything. I remember this is 20 years ago. I'm like, yeah, okay, that's cool. I'll try it. And you're like, I don't know. Like you give up after 15 seconds because we're all lazy, right? And we should be, right? Okay, so that's on us. So we have the person imagine writing words, for example, hello world. And then we record from neural activity in the hand and finger region of the brain, the so-called hand knob area of the precentral gyrus, so premotor and motor cortex counterparts in monkeys. And then our algorithms will, once a letter is complete, once you get through the H, then an H will pop up. And you see here, he's not quite done with the W, so the W has not yet popped up. Okay, so it's character by character. And you can write any letter you want, and that constitutes, very importantly, an open vocabulary, an open dictionary. Okay, that means you can write any word you want, period, right out of the gate. And this has been a vexing problem in the field when you use EEG or ECOG, because the neural signal to noise ratio is not high enough to support doing this. Instead, what you do is you try to detect words, and then you say, okay, given this neural evidence, which one of a small dictionary size, which one of 50 words was I thinking? And you can do reasonably well, but it's only 50 words. And an average 8th grader typically has 10,000 to 15,000 words at their disposal, okay? All right, so here are our 2 electrode arrays on an MR image of our participant sitting on the homunculus right about here. More on this in a second. We're deeply questioning the homunculus, okay? And here what we did was we said, let's start off by asking him to prepare to write a letter. So the computer would say, okay, let's try A. You wait a little bit, you say go. He attempts to write the letter A. Now, what does attempt mean? If I ask you, what are you thinking when you move? Right? Most of us will say, I don't know. I just move, right? It's not really a conscious act, okay? All right, so then how in the world do you tell somebody that can't move what to do? Well, there's a few magic words. You could say, please imagine writing, or please attempt to write, or please envision writing. And there's been a rich set of studies through the years that we and others have done. And attempt seems to be the best word that we think creates activity that's most similar. You can't really go wrong. They all sort of work, but attempt makes sense to people and elicits really robust activity, okay? So attempt to write the letter A, and then we move on to the next letter. We do this a few hundred times, so we get many examples of each letter, and then we move to words, and we say, okay, let's go through a bunch of words, okay? And then once we have the words, what we can do, and more on this in a second, you can chop the words up into individual letters. And then the real trick is that you can perturb those letters synthetically so as to create augmented neural data, which is essential for getting enough data to really train machine learning algorithms well. But you have to be very careful how you augment that data, okay? Now, that's on a calibration day. So we can walk in. We do need 1 day or a couple of hours to do this, train our recurrent neural networks on some big computers, okay? And then what comes back is a very lean, simple recurrent neural network that runs in real time. The next day, you don't have to retrain it. And you can not retrain it for a week. Or after a week, or even if you want to the first day, what you can do is you can only take like 10 minutes and go through some of this to recalibrate. You can fuse that back with your already existing knowledge to augment your RDN, and you can maintain performance indefinitely. Okay, so it's not a training burden and it's not hard. So how well can we do? So let me show you something that is not a main part of the system, because we are not trying to reconstruct handwriting, penmanship, right? We're not trying to restore the ability to write calligraphy, okay? We don't care about the exact A that was drawn. We are into classifying. We simply want to know, is it an A or a B or a C? But what I want to show you is decoding the velocity of that attempted pin tip as a function of the neural activity with a simple linear decoder. And what pops out when you look at the average across several different A's and B's and C's are things that look rather like the letters, okay? Now, that's what we expected, okay, because that's what he's attempting to do. It's a nice confirmation. But the real point is, look how different an A is from a B, from a C, and a D. And if we can decode it and it looks that different, we better be able to classify it, okay? The one thing I haven't shown you is how well it can be done on a single trial, though. This is trial averaged, meaning you can take 10 repeated A's, average that activity together, which beats down the noise. So everything about real-time and neural populations in the body or in prosthetics is about noise, okay? But not everything, but you know what I'm saying. All right, so here's that plot. So what I'm doing is I'm showing a two-dimensional space you can think of as principal components space, which captures the first two dimensions of this 192 channel simultaneous recording. And it does so to capture the largest part of the data variance. This happens to be a nonlinear technique called t-SNE, but it looks very similar using a whole variety of different methods. It's not about the method. So let's look at the letter G. So this is where our participant wrote about 15 different G's, right? He's writing the alphabet in a random order, and oh, there's a G. There's a bunch of other letters. Oh, there's another G. So in one day you get 15 of these, for example, and you put a point down every time the trial completes, and you're looking at dimensionality reducing that neural activity. And the key point here is look how similar all these red dots are, where they're color-coded red if it corresponded to an attempted G. Here's an S. You have a lot of white space in between. That means you could put a line there, and that means you could classify this G from this S perfectly every time. Okay, now that's never true, right? You do it enough, you're going to get one thing that's confused. There's always going to be errors. Now let's look over here at the greater than sign, which we use as the space bar. Okay, it's a little easier than hitting a space. An X and a Y. Well, these look totally overlapped. It looks like you're totally in trouble. You can't tell those two apart. And that also sort of makes sense because look how similar an X and a Y are. I mean, you just don't have this little tail here on the Y, right? So everything's sort of lawful. It sort of makes sense. Mercifully, this is only in 2D, but the data actually live in a higher dimensional space, typically around 15 or 20D, okay? A little higher than for reaching arm movements for aficionados, okay? And so if I take these data and you just give me a third dimension, and I should do this, you know, to rotate a movie around, you'd see these clusters split apart. So it's also easy to tell X and a Y from each other. It's just this viewpoint that makes them look confused, okay? Now, you can't do it perfectly, but we can do it with 94% accuracy. Okay, so 94% of letters that are attempted, we can tell you exactly which one of 26 letters or 5 special characters, commas, spaces, and so forth it is. So at this point, the game is afoot. It looks like it should work. This is all offline, meaning you record the data, you look to see how well you could do. Now let's go build the online system. So what we did was we said, okay, our data are going to be flying at us, 192 electrodes worth through time. We'll have a black tick every time there's an action potential fired. Okay. We're going to look at a brief time bin. In that bin, we're going to count the number of spikes that each channel, each electrode had a spike on it. And that'll give us a column vector of data. And this will just start running forward in time. And what we need to spit out the other end of our algorithm are these letters that we can render on the screen. Okay. Again, we're not going for penmanship. We're just going for the actual letters. Okay, like typewritten. So long story short, recurrent neural networks, which have really revolutionized speech processing, because they're really wonderful at dealing with time series data, just like convolutional neural networks have been wonderful working with the ventral visual stream at classification. If you train a recurrent neural network, what we can get out are character probabilities as a function of time. In other words, up pops the probability close to unity for the letter T, and then N, and then E, and so forth. And what we can also do in parallel is we can get a signal, just a binary signal that goes high when there's going to be a new character and low in between. So these two together allow us to simply put down a threshold here and also use this information to have what we call the raw output. Now, the raw output of these letters is TNE paper, okay? Well, I don't know what TNE paper is. I know what the paper is, okay? This is an error, obviously. So you have two ways to go here, okay? Probably more, but two that we've explored. One is, if I send Juan an email and I make a single character typo, I may not even bother correcting it because I know Juan will be able to understand it, okay? So that's not an excuse. That's not to say, oh, that's good enough, okay? But it is important to keep in mind, you can still get the meaning pretty well because there's so much redundant information in the language, okay? That's number one. Number two, not good enough, okay? There's no reason in the world we should be making this silly little error, okay? Why is that? Well, because we all, even Google Mail can fix this for us, right? So auto spell checkers, word completion, all these additional, they're not neuroscientific, but they are, of course, very important engineering and algorithmic things. So what we can do is we can take a language model, which, welcome to 2021 or welcome to 5 years ago, but not 10 years ago. You just pull down a nice language model like Caldi. It has 50,000 words in it. You know the exact probability that one letter like H will follow a T. That's, in this example, 0.9, whereas an N is only 0.1, okay? So there's no way that N is correct. So when you traverse through the optimal path, which you can get to with the so-called Viterbi algorithm, you can actually automatically repair it. So the way that this would work is he's typing along T, N, E, space, paper, and then right about here, when the word is completed, the algorithm says, that's not T and E, that's I'll just change the N to an H. Now, it better work better than my iPhone works, right? Because I don't know about you, but I always turn off my auto spell checker because sometimes when it swaps out a word, it mutates the meaning so badly, the receiver can't even understand it. I may have said something like, totally embarrassing by accident. Okay, so but that's modern machine learning, and that will get better, okay? With this, we can drop the error rate by an order of magnitude from 5% down to about 0.5%. And that's what's shown here. So if we look at the characters per minute as a function of the days, we can see that we see improvement because he's getting used to the system some, okay? And we wanted to plot the first 5 days that we used because we really like focusing on how rapidly people without prior training can use it. Because again, if you need to train a person who has a very debilitating disease for too long, they don't do it. It's not worth it, okay? So this is really important, okay? We're able to get up to about 90 characters per minute, okay? That's about double the previous world record of brain-computer interface typing, which happens to be from Chetan Pandaranath, wonderful postdoc, and Paul Nuyajukian, wonderful postdoc in our group. Chetan's now at Emory, Georgia Tech. Paul's on faculty at Stanford. And the language, the error rate, the so-called character error rate, how many characters were wrong, bounces around 5, okay? And that's raw. And then with that language model applied, we go down to 0.5. Now, importantly, we have not run that language model online. We only did that offline, okay? But there's absolutely no reason it doesn't work online unless that word swap distracts him, for example, okay? Anyway, that's next up. So those are experiments that we have underway. So let me show you 2 videos and then move to speech. So what does the whole thing look like? Okay, well, here is the first time I'm showing you what our participant, where his designator number is T5 to protect anonymity, even though he uses his name publicly. We as investigators are just hyper overtrained never to say the name. And what you're seeing here is him sitting up in his wheelchair, which he needs assistance to do because he can't move either arm or legs or hand. Okay. And then 2 of these electrode arrays are implanted both on the left side in that, as I showed you on an image of his brain. And these 2 connectors, just the metal part, not this big gray box. That doesn't stay on him all the time. That's just when we're running sessions with him. Those do not represent where the electrode arrays are. The electrode arrays are right here. There's just a wire come out here, so you space these nicely, okay? And this is what all has to go away. I mean, good grief. You have a big preamplifier and cables. Okay, this is another classic. The medical device industry trails commercial electronics probably by 20 years, okay? It should be fully implanted and wireless. He's looking at a sentence on a screen and we ask him to copy type it. And why do we do that? Well, we also just ask him to say whatever he wants. We've absolutely done that. That works just fine. But you see, that's not as fast as he can go, and we want to measure how fast he can go. Why? For the same reason that when I was young, at least, and you were in school and you learned how to touch type, you never were asked to just type whatever you want. You had to copy type. And that's because it takes you time cognitively to think about what you want to say. And that's an uncontrolled variable. Okay. But it works. But here we ask him to copy type it. So the green just says go. Okay. And let me play the movie here. And what you're seeing are these letters just seemingly magically popping up on the screen. Okay. And the movie is a little jerky on my end. I don't know if it is on yours, but it's absolutely smooth in reality. Okay. And it's at 90 characters per minute. Now, the key thing, I should show you the movie too, but for the sake of time, I'm not. You see, we have tons of cameras around everything. If you look at his hand, it does not move. Okay. So this is a supplementary video. Okay. Or at most, if it moves in a slight twitch way that's not at all correlated with handwriting. Okay. So he can't move. He's attempting to move, and that's the performance. Now, one last video here is the current system that I was just showing you up at the top, where he's going to copy type, "You must be the change you wish to see in the world." Okay. And this is going at about 17 words per minute, 95+% accuracy. And what's down here at the bottom? Well, it turns out this is the same participant from 3 years ago when we ran this point and click design, okay, which used new Kalman filters and new neural HMM decoders, so you could move a cursor on this keyboard and hit a click signal. And this is Chathan and Paul's beautiful work, and it's 8 words per minute. So we can play them side by side, and not just so that you can see the performance, because you'll see this go twice as fast, which is, fine. It's a pretty movie, okay? But the real thing is this much higher performance was recorded on the same arrays in the same person 3 years later. The signals are definitely not better than they used to be. They're actually rather similar, but we can do a whole lot better because of the algorithm. So the point I'm trying to make is, again, these electrode arrays are not great. We all wish for better, but they don't just fall apart, okay? So here you can see how he can move the 2D cursor on the screen and type. Okay, and here you can see him typing the exact same sentence, right? Because we just replicated it. We just dug up this movie from 3 years ago, okay? And that's the performance improvement. All right, so now in a much shorter vignette, let me tell you a little bit about speech, okay? And let me tell you what I'm going to tell you about speech. So 2 points. First is we're finding speech-related motor signals in an area that you would never predict to find them, okay? Meaning we can decode the phonemes and the words you wish to say from the hand and finger areas of the brain, violating any even loose adherence to the homunculus, okay? Second, we have not built this system in real time. That's what we're aiming to do, but we believe we've done the preliminary work, just like I showed you in handwriting, to make us believe, we've become convinced that it should be fully possible. And the, 3 points. And the third point is that these systems have definitely been built. We're highly inspired by our friend and colleague up the street, Eddie Chang at UCSF, who uses ECOG grids, Mark Slutsky, and so forth. So you can think about words, speaking, decode them, that's great. The different thing here is that we're recording from individual neurons for the very first time. All right. So this is work by Sergei Stavisky, who's just about to accept his faculty position. Frank Willett, who you saw before, Guy, who's a wonderful graduate student, and Shaul Druckmann, who's a wonderful colleague at Stanford on the computational side of things. So we're definitely inspired by the seminal work by Eddie Chang and Mark Slutsky, who put these, you can think of them as just sheets of plastic, if you will, with a grid of electrodes that really broadly covers an area. And this is done for a couple of weeks when people are in the hospital that need to have this done anyway to understand where epileptic seizures start, because this is how you plan tissue resection that neurosurgeons perform right after this mapping is done. But people are sort of just sitting there waiting for an epileptic seizure to happen. And so they're very happy to actually participate in other experiments like speech and so forth. And that's what's really been capitalized on beautifully. Now, as I said, here we want to look at individual neurons because we do believe that individual neurons are where the most information is, okay? But the trade-off is going to be we can only record from tiny little parts of the brain as opposed to huge swaths of the brain. So which, I don't want to say which is better, they're going to complement each other, but does this also have a role? So our participant listens to a couple of beeps that he hears on a speaker in the room. He hears the computer say "ba," for example. A couple of clicks happen, and then he has to say "ba." Okay, and we can record that audio track. Now, he can still speak, okay? Now, hopefully he will not lose his ability to speak because it's a spinal cord injury. But unfortunately, many, many people do. This is a classic thing that happens in late stage ALS or Lou Gehrig's disease, okay? And this is just a golden opportunity because he can still speak. And the reason we could do this line of work responsibly and ethically is if the electrode arrays were already implanted in this other area for this other purpose. So we basically took a giant gamble that, you know, it's not that hard to try this experiment. Let's see if we happen to find speech stuff in this area, because if we do, that's weird. Okay. So here are the two electrode arrays. And now we're going to really spike sort because I want to be sure that we learn and share what individual neurons are doing. Okay, then we'll move back out to looking at populations of threshold cross neurons. Okay, but here's an action potential from one neuron. Now these are repeated trials for silence and 8 different phonemes. Okay, and you can see different activity patterns, but if you look in a so-called trial average PSTH, a peristimulus time histogram, you can see that the red and the green, ga and ba, really elicit high firing rate, whereas silence is far lower, and blue and purple in here, pa and sha, much less. And these are confidence intervals in the gray bands, okay? Now, here's another neuron. okay, which has a different pattern. It really likes K, okay? So the point here is that different neurons are going to have stronger responses for different phonemes, number one, okay? And number two, a given neuron is going to respond to many different phonemes. And number three, there's some weird rich structure here that is a little hard to understand, but these are in areas that project to the vocal tracts and the lips and the tongue, okay? So this is a motor control problem. And in that domain, as Juan was introducing, this is what drove us many years ago to start thinking sort of beyond what a single neuron does, because then you tend to wind up telling sort of just so stories about what each individual neuron is representing. Instead, we say, listen, I don't know what it does. I give up. I just declare a failure right away. Go measure a bunch of data and then go look to see what the data say, okay? So this looks very familiar because it's complex and heterogeneous data. Now, if we go look at all the electrodes that are statistically significantly tuned, shown in blue, when he says I, you see that it occurs across the array. And that pattern is partially overlapping, but also different for all the different symbols. And on average, or the median, is 4. So the median number of syllables you get statistically significant tuning on is 4. Okay. Does this hold up in a second participant? Yes. The data are not quite as clean. Welcome to real world neuroscience, right? Okay, but it's there and it has all the same trends and patterns and statistical significance. Okay, and now what about this single trial neural activity again? So this is the same plot that I introduced in handwriting, where we can now ask in this 2-dimensional t-SNE space, how different do repeated trials of silence look, or SH look, or K look, when we look at the activity across all of our electrodes? This is a population neural activity projected into lower dimensions, okay? And the answer is that not only for those 8 phonemes where we could do a reasonable job, but more importantly, when we record 420 different words in large corpus of words that are read back, where, for example, the word word can be broken up into its individual constituent phonemes, okay? We can then go ask, if we speak one of the different 39 different phonemes in English, and we plot here the phoneme that we decode, how well do we do? Now, if you see a totally dark blue diagonal band here, then we're doing perfectly. Whatever phoneme we speak, we also decode. Great, okay? Clearly, that's not the case here. Clearly, that's not the case here. In fact, it's even worse than that, okay? While we absolutely do see a diagonal band, and that's good enough to decode 29%, more on that in 1 second. If you look at the error structure, the errors are sort of uniformly spread across all of our phonemes. So when you do make a mistake, it's not like you're only making like a 1 phoneme off error. So this really has sort of unrescuable error structure to it, if you will. Okay. Now, what about this 29%? I don't know, what does 29% mean? You know, it's a number, but what do you reference it to? Well, clearly we know that 100% would be awesome and 0% would be horrible. And 1 over 39 is chance, like 2.5% or so, okay? Well, 29% we believe to be not good at all because we like things in the 90s, okay, number 1. Number 2 and 3 are really the key points. The second point is we're in the wrong area, right? Our electrode arrays are not in the heart of speech motor cortex, or just interior to that, inferior frontal gyrus, which contains Broca's area. That's where you'd want to put arrays. that's where we will put arrays. Now that we have data to support that we can do this in people, that is ethically motivated and FDA approved as of a couple months ago. Okay, so when we put the arrays in our next participant in the correct area, we think we could do a whole lot better. The third point is this 29%, which is like, not really that great. You could do something with it. Interestingly, this is already in the wrong area as good as ECOG does. Okay, and that's really speaking to, it's nothing we did, that's just speaking to the power of the information present in individual neurons. Okay, so next steps, what we'd like to do, as I just said, is put electrode arrays not here and here. This is where I showed the two arrays are, but for speech, we want to put them far more lateral in the classic speech and face motor cortex areas, and then in the inferior frontal gyrus Broca's areas. Okay, so that'll be our next participant. And then for the handwriting work, where again, we think that we'll be able to pick up finger and hand related movement all along the motor strip, so we'll sample that more, and then also bilaterally. So we'll put a couple of other arrays behind here. So all of a sudden you're starting to see lots, a lot of little colored squares show up here, right? Why is that? Well, that is the result of Lee Hochberg's great vision, who started the BrainGate clinical trial 15 years ago, which we joined in about 11 years ago, where we've laid down systematically over the last 15 years the safety profile as part of the clinical trial that everybody is now more comfortable, including the US FDA and ourselves, most importantly, that we can safely put in more electrode arrays, including in eloquent cortex, including in areas which people are still using to speak, but in ALS participants, which is our target population, they will soon lose the ability to speak. So that's why it's justified. Okay, so in conclusion, I've described a new and different and weird brain-machine interface called, attempted handwriting. And there appear to be really robust neural representations of these highly dexterous behaviors in our participant even a decade after spinal cord injury. Okay. Demonstration of a real-time handwriting BCI. This goes much faster than before, more than double. Okay. Now, a couple points I didn't go into. I could, easily have another 10 or 20 slides on these things, but the BCI is self-paced, the point that I made early on, but I didn't dwell on. The point here is that what we do when we're learning is that we time stretch or compress, okay, called time warping, neural data, so that no matter how quickly or slowly, within reasonable range, a letter is written, we already have the model tuned to be able to decode that. And the behaviors that are time varying may be easier to decode and lead to higher performance is, with more time, that's probably the number one topic I would want to describe, which is quite simply, and I can summarize it for you. Wait, why in the world is handwriting doing better than moving a cursor on a screen? Moving a cursor on a screen in just different directions and typing seems so much easier. You should be able to do a whole lot better than this whole complicated write letters and decode them and classify them. Well, it turns out that if you think about what you can do in 1 second, in 1 second, you can easily write a letter. In 1 second with a cursor, you can easily move in one direction, or maybe ultimately you can move in all the directions that write a letter, but you're basically making straight lines. So what a letter has is that you are visiting a wide range of different directions, okay, because you're changing that so rapidly. And that kicks up the so-called dimensionality of the data. And that's what makes it easier to classify. So in the paper, this is figure 4, where simulations and analysis and everything show that that's really what's going on. So that's really the core science insight that we built everything off of. On the speech side, we've surprisingly found speech-related motor activity where we should not, which is centimeters away in what we thought would just be hand and finger and maybe arm, but not speech, meaning facial muscles and vocal cords and stuff. Okay, yeah, sorry, not vocal cords, but articulatory tract. These single neuron signals show a lot of promise, we think, for speech. Okay, and finally, the very most important slide I ever show, which highlights the wonderful team, the wonderful students that I've tried to point out along the way, and postdocs. This is Lee Hochberg, Jamie Henderson. A special shout out to Jamie. He is my partner. I would say partner in crime, but that has a bad connotation. My close collaborator for the last 10 years. He's the functional neurosurgeon at Stanford who puts in deep brain stimulator electrodes by day and then does this by night, shall we say. And it's really these types of interdisciplinary collaborations and then across institution collaborations where Lee is at the Massachusetts General Hospital, Brown University, that it takes to do these very fun interdisciplinary human-oriented projects, because they're an awful lot of work. And finally, wonderful, just couldn't be prouder of the people that have come through the lab. Thank you very much.
We would all be clapping, Krishna, but thank you very much for the really nice talk. There's a bunch of questions. So, well, there's one question, someone raising their hand. So basically, if you raise your hand, I will unmute you if you want to ask Krishna in person, or if you are shy, have bad connection, I can read your questions.
Yeah.
So
should I release the screen? Does that help? Or it doesn't matter? I don't know how many people would show up. Is it easier to see each other, or?
Yeah, there's not a lot of people with their cameras on, so we can leave the screen in case.
Oh, okay. Yeah, in case we need to reference it. Yep.
Yeah. Okay. So there are already a couple of questions on the chat, so I will read them for you, Krishna. One is from Itamar Landau, which I'm sure I haven't pronounced correctly. So they are asking whether you can say a few words about what we may learn from these recordings and algorithms about how the brain controls movement.
Oh, yes. Okay. So to rephrase the question, if we're decoding neural activity to drive prosthetics, does that teach us anything deep about the neuroscience of how maybe the downstream cortical area is decoding it and everything? Okay, so I think so this question often comes up, and it's a great question. And the way I think it's the way I like to answer it, which is, you know, the honest way I think about it, is no. Okay, and I'm going to overstate it that way. Then I'm going to back out a little bit. Okay, so the answer is no. Okay, now why do I say that? And why do I happily say that? Well, the way we've always thought about things in our own lab is that we are very happy to do neuroscience. We're very happy to do engineering, brain-machine interfaces, and the two worlds never need to meet. Okay? It's much more important to try to do good neuroscience and do good engineering and just be happy with that. Okay. Now, why is that? Okay, and this gets at the heart of the question. The reason is that I guess I've never wanted to accidentally try to work right in the middle, trying to do both neuroscience and engineering on any one project and coming up short in both domains. Okay? All right. Now, with that disclaimer and that said, do we learn anything at all by what we do? Of course. Okay. But the first answer is no, nothing at all. Okay. Now, what do we learn? Well, if we can decode information, we know that that information is there. There's no denying that. And decoders have been used, they were originally sort of innovated for brain-machine interfaces, but they've been used widely throughout neuroscience and fMRI, and they're widely used as a way to bound the amount of information. You set a floor. You know that there's at least this amount of information. There may be more. But second, you never know if that information is used, right? That's a separate question. It's there, but is it used the way you think? And third, the way you extract it with a machine learning algorithm or a statistical signal processing algorithm and so forth may or may not have any bearing on how the brain actually decodes it. And so those are more the no answers.
Okay, great. I might say like Krishna has a few reviews that summarize their work, showing like this research on motor control. I guess that you would have fun reading. There's a lot of questions. I want to encourage people again to raise their hand and ask in person, but in the meantime, I will read the next one, which is from Jiju Yang. Again, pardon my Mandarin. So yeah, like their question is, how does the electrode affect the function of the neurons? So I guess this is biocompatibility.
Yeah, that's a great question. You know, I think most of us think, or at least want to think, that the presence of the electrode does not alter the activity at all. Okay, that you're just sort of sneaking up and peeking at the natural neural activity. And I actually don't have any reason to think that that's not the case. But also what is the case by definition is you're putting an electrode into the brain and you're definitely killing cells and changing the local connectivity to a certain extent, inevitably, because you're putting something physical there. It's not like optical imaging where you're truly not touching anything, okay? I mean, there you're altering it genetically to be a calcium reporter, but okay. But third, it is interesting. A lot of people do talk about this from time to time, because when you put an electrode there, you are changing the local electrical environment. So, you know, from an electrical engineering perspective, or just, you know, just the classic electrophysiology perspective, if you actually think about potential field lines and current flows in the extracellular space, the ionic currents and everything, there's no doubt that you're changing that too, because you basically put an electrode that you have a certain potential at. Okay, but the way the field mostly thinks about it is, and I think this is largely correct, it does not alter that activity. Yeah. Sorry, I should say one more thing. That's if you put an electrode in for like 1 day. If you leave an electrode in, like these, a chronically implanted electrode array, then you need to also think through all sorts of immunological responses, inflammatory responses, glial and astrocyte cell dynamics, you know, what kicks in, what may wall off the electrode if it's maybe less than fully flexible, so the brain always senses the presence of this rigid silicon shaft that can push cells away. So there are all these alterations that happen, probably much more for chronic electrode arrays. Yeah. Okay.
Perfect. I see some people are raising their hand. Lee, I will let you ask Krishna a question in a minute. Before, there's Siegfried Ludwig.
Hey, Lee.
I'm going to read this.
We come to London to see each other in Chicago, and yeah.
Lee, you're muted, but it's fine because I will read Siegfried's question before you to follow the order. Yeah, like Siegfried is asking, could you elaborate on the idea that the visual cortex has the movement intention and premotor cortex translates it into action? Which I think may be a misunderstanding, but I don't know what you want to comment on that, Krishna.
Yeah, well, you know, so if I understood the question correctly, maybe visual cortex has the intention and motor cortex implements it or elaborates it.
Yeah, yeah, that is kind of the question.
Yeah. No, it's a good question to ask. And, you know, as with so many things in neuroscience, it's awfully hard to be positive that that's not happening, I suppose, but that's clearly not the way it's mostly thought about. Mostly visual cortex is thought of as a sensory area, V1, meaning primary visual area. It's sensing information. And then it's throwing it forward in the dorsal stream on the top of the brain into parietal lobe, where you're then maybe calculating your error vectors, how you want to move your arm. And that goes to premotor areas and motor cortex, where you plan and move. But it deserves to be said that the really intriguing levels of motor modulation that are being found in visual cortex in mice and maybe, probably other animals, even humans, is humbling, right? It's like, it's a sort of every 10 or 20 years, you question your very existence and way you think about science. So it's a little hard to say. It's also worth saying that so many things are reciprocally connected. A projects to B, B projects to A, that there's a lot of correlation in many areas that's intriguing. How can the brain handle that? Why didn't it go to the effort of eradicating that? Or maybe it's using that. It's sort of like the speech related signals that we're finding in the hand knob area. A common question, one we've asked ourselves, we just don't quite know what to do with it yet or at all, is, hey, look what I'm doing. I mean, we all talk and move our hands at the same time. And there's probably some level of deep coordination that a community that I probably don't know about maybe studies, right? Because it's all part of, this is nonverbal communication, right? Part of what we're missing in this year of Zoom, we can barely see each other's hands, let alone body movements. So maybe it's important that an area have both hand and speech information. Could be, we don't know.
Okay, great. Liz, your turn.
No.
Great. As I said, yes, Krishna, it's certainly nice to see you here in London. And I'm going to take the opportunity to change my question from what I posted in response as a result of the earlier question about whether the electrodes change the brain, damage the brain, whatever. Another intriguing possibility that just occurred to me, we all know that brain-machine interfaces back from the very first ones that were just used to move cursors cause the modulation of those neurons that are being used for control to change. The brain actually learns which are the important neurons. So presumably, as you begin to decode speech from the brain, you're going to be affecting the way those neurons, in fact, are modulating. Have you given any thought to that?
Yeah, that's a great point. I mean, that's another whole fascinating dimension that, many wonderful colleagues have worked on. Eb Fetz is, one of the pioneers, Jose Carmena, and many others in the field, of course, too. And so just to unpack it a little bit, what Lee is saying is if you take out the activity of just one neuron, just to keep the example simple, and you use it to move a computer cursor up or down on a screen, the higher the action potential firing rate or emission rate, the higher up on the screen it will go. And you can train monkeys and presumably people to control that. And what you find is that through time, plastic and adaptive processes kick in, and you actually change the relationship of the natural neural activity that controls that, okay? And if you look at this with electrode arrays, as for example, Jose Carmena has done, you can see that through time, you get this beautiful adaptation to make things work better. So there's definitely the possibility. There's definitely the possibility with speech. We have not thought deeply about it at all. You're the first to sort of really encourage us to do so. But I can take the opportunity to say one more thing, which again comes back to Jose, and this dates back about 5 or 6 years now. We wrote a piece in Neuron in 2014, when invited to do so, in direct response to the following. I was giving talks about brain-machine interfaces that work pretty well right away, because we did a so-called biomimetic decoder that Lee's a pioneer of and knows all about, okay? And not at the EMG level, like we'd really like to, but at a kinematic level. And Jose, his beautiful line of work is saying, listen, let's not use the best decoder. Let's actually use a decoder that there's error on the screen, okay? And see if the animal can learn to adjust for that. And this is highly related to wonderful work that Andy Schwartz and Rob Kaz and Beata Jarsikowicz did at Pittsburgh a few years before, where they intentionally rotated so-called preferred directions and watched it adapt. And so Jose would get the question of saying, hey, once you do this, how does your performance compare to what Krishna has been saying? You know, does it get up there? And the answer is no. But it seems like we should be able to put the two together. I would get the questions. You're not talking about learning or adaptation at all. What's going on, right? Surely you see that. And the answer is no, actually we don't. We've looked for it, but the reason we think is that because the systems work well enough right away, there's very little pressure on the animal to change. But if we put them together, and this is what we outline in that piece, it ought to be possible that we don't have any great ideas, but it ought to be possible in principle, at least, to put the two together to engage learning to increase performance to even better than you'd otherwise be able to do. So in your question, Lee, for speech, I think a similar thing might happen. If we're able to decode it well enough, then we may not see much. I don't think we'll be able to decode it that well. I bet we do see adaptation. And, you know, this is a huge story, as you know, but I'm just unpacking it for the group here. A big part of the story of cochlear prosthetics, cochlear implants, where you stimulate, you don't get natural sound, but over days you learn to interpret it much better.
Cool. Okay, I'll keep reading questions then. So Etienne Bourdet, I don't know, Etienne, if you want to unmute yourself, or I guess I will just read the question. Yeah, so Krishna, Etienne is asking, so the speech generating system could go indirectly over writing. So I guess over the writing system. How would such system compared with direct speech intention detection?
I'm sorry, could you just repeat the question?
Yeah, I'm trying to unpack the question myself. So Etienne is asking, the speech generating system could go indirectly over the writing generating system. How would such system compare with direct detection of a speech intention?
Okay, well, let me, we may need to ask for some further clarification. Let me try to.
We can maybe wait to see if Etienne is around. Etienne, can you?
Well, let me.
Okay.
Yeah, well, I mean, you know, at present we view the two as separate and in parallel, but maybe the question is if you wanted to use both in combination or so forth. And that there is a relevant study that we did. So with the attempted handwriting, that works the way I described. And then with the same area, when you are attempting to speak, or in this case, he can actually speak. So it's actually, you know, attempt and actually do speak.
Mm-hmm.
We can decode that. Okay. So the question could be, I don't know, but it's a good, fun thing to share anyway. The question could be, well, wait a minute. What if he's talking while he's attempting to handwrite? Are you going to get both sets of that activity? Is that going to confuse these systems? Okay. So we don't know. That could happen, and would need to work on how to sort that out, because if a person can talk but wants to use an attempted handwriting system, you wouldn't want to say, well, you can't talk. You're using a handwriting system. You can't talk. We'd like to algorithmically separate that. And the possibilities are that either when you are attempting to handwrite, if that's the more dominant signal, it could suppress your speech-related signals. I think that might be how it's working. I'll explain in a second. Or the other, sort of actually more interesting way for those of us, Juan, certainly you and Lee, you and others that think about dimensionality, be very cool if they were coded in different, orthogonal subspaces, because then it's very easy to say, oh, okay, well, speech is in these dimensions and handwriting's in these dimensions, and there you go. So we had the same question when we first discovered speech, and we're using not attempted handwriting, but the 2D cursor control. So we did that experiment, and that's a paper from a couple years ago, where we said, wait a minute, how are we finding speech here? Because surely we would have noticed with that hundreds of hours we spend with our participant, that if they're trying to move the 2D cursor, that if they're also talking, the cursor performance just falls apart. You know, be like, did we not notice that? Okay, so when we went back and we did careful studies and said, okay, just use the cursor, or now just use the cursor and try speaking, the answer is crystal clear. What happens is that when you are moving the computer cursor on a screen, in other words, imagining moving your hand moving, it quashes, it really suppresses the speech-related activity. Okay, but the second you say, okay, stop using the arm, attempted arm movement to move the cursor, now just speak, you know, the speech information is there. So the interesting thing here, and we went into this some in a Cell paper last year by Frank Willett as well, where when an area seems to have multiple effectors involved, because the fuller story to this is that in this hand knob area, we're not only finding arm and hand movements and speech movements like I've described here in this talk, right? But when you go try the ipsilateral arm, meaning the other arm, and legs, contra and ipsilateral legs, this area modulates for all of that. Now it's strongest for the contralateral arm. So that's why it makes sense with the homunculus. If you're sitting there, as Penfield did, you know, back in the 40s and so forth, and you microstimulated, totally understandable that you get arm movements. That's where most of the activity is, okay? And with ECOG, when you're recording from, you know, thousands of neurons under each electrode contact, it makes total sense that when you're up here in this dorsal motor area, you get arm and hand. Makes total sense. And it's much more lateral that you get the speech area. But when you look at individual neurons, it is absolutely multimodal. It's multi-effector. And so what's interesting is that when you start pairing movements, it seems like the dominant movement wins out and suppresses the other movements. So that could be a benefit because we didn't have to do anything to make a 2D cursor system immune to also speaking. That's why I'm guessing the same would be the case for handwriting. I don't know. And it could also be a con because we can't do the two simultaneously, it doesn't seem. You know, probably we could, well, depending on how strong the signal is, maybe we could figure something out, but not at first blush. It looks like it's really squished.
Thank you very much. So unfortunately, the system didn't allow me to go from my computer, but coming to the neighbor, it's okay.
No problem.
Thank you very much. So it's explaining well. Yeah, so basically, I would be interested to understand whether we could use different representation of language, I mean, of speaking. So some visual representation and a more phonetic representation at the same time. That's what I would like to understand.
Yeah, so we haven't, you know, done any work along those lines. Yeah. But you know, just to sort of reiterate, it may be more than just a motoric signal, but we are very much viewing these signals related to language that we're recording as purely motor. Okay, meaning, so another part of this study that I did not share, I didn't have time to, regards, well, okay, hey, these same neurons that are modulating for different phonemes, if you're claiming that they're motor related, then show me two things. First of all, show me that the activity is far less when you're just listening than when you're speaking, right? Because you should find it when you're generating movements. That absolutely holds up, okay? So it's speech production related, not auditory. You know, our internal joke is, did we accidentally implant auditory primary cortex, right? So no, okay. Second is, now I've lost my train of thought, sorry. Oh yes, So if it's motor related to speech, then when you're saying a word, you're contracting your lips, moving your tongue, your soft palate, your whole auditory tract, okay? So what if you had the participant not say anything? In other words, don't exhale and produce the sound, but go ahead and systematically move the lips, move the tongue left, right, up, down, everything. If it's motor, Krishna, you should still see strong modulation. And we do, okay? So I think some of the questions you're getting at are more related to higher level language, semantics, like you were saying, or meanings of words and these types of things. So that's where we're very excited to be heading into recording from this upstream area, inferior frontal gyrus, Broca's area, as we all learned it from the classic textbooks, related to speech production, right? I always have to remind myself, Wernicke's is speech recognition, Broca's is speech production, okay? And then it projects forward to the lateral motor cortex, okay? And that's the counterpart where we're recording. It's just that we're not recording in as sweet a spot. Yeah. Then we might be able to get at, and what we're very much hoping to get at, is more like we think of, higher level area like supplementary motor area is for reaches, where it deals with sequences. And then the primary motor cortex that reads it out and actually generates the movements. There we might be able to start getting into some higher level language type concepts.
Thank you.
Okay, I'll take back control of my computer to read the next question. Yeah, so this is from Chris Rowlands. Chris is asking, what do you think that, and it's actually related to your conversation with Etienne. So he's asking, what do you think the true latent representation of a written piece of text is in the brain? Is writing letter by letter efficient if the brain really conceptualizes the text in terms of words, or perhaps more likely linked to linked concepts? Sorry.
Yeah. So first of all, I should make I think what's probably very obvious, but just to state it up front, I am not a speech expert. Okay. What drew us into this at first a little bit reluctantly, but a couple years ago, was just the super tantalizing motor control problems related to this. Okay. And, I think we all have a part of ourselves that wishes that we work on birdsong. I do. I'd love to do a mini sabbatical in birdsong. Okay. How do you crystallize these unbelievably complex motor sequences that are done with incredible precision and so forth? Okay. So it's such an alluring motor control problem. Okay. And, the number of times I wished I could talk with my monkeys, but I couldn't, this is, again, something too alluring to do with people. Okay. So with that said, we do view everything that we're doing, again, at this very low level. And when it gets to, what is the brain really doing? Is it really thinking about letter by letter? Is it thinking word by word and so forth? I'm afraid that, to the best of my reading and conversations for the last few years, we really don't know. I choose to view that as a huge opportunity. We can all introspect. And I think we're all very much guided when it comes to language by introspection. And this comes up in numerous conversations I've had, which is we all hear this inner voice, right? This inner voice. We think that we think largely in and around language. I think we all believe that word level, probably word level, is maybe sort of the natural, whatever natural means, but chunk. And then you just push it off to some, motor control system that actually creates that sound. But what you as a person are really doing is thinking in terms of words. That's where you manipulate concepts. That's where you form ideas. That's how you retain information. But I don't even know what that would look like if I had it on the tip of my electrode, right? Again, I view that as exciting, but also terrifying because, you know, maybe it's a fool's errand to even try to tackle some of this. So so what our strategy is we're very interested in it, but creep up on it. Okay, start with the motor system and then keep pushing up the hierarchy, and that's absolutely a classic playbook, as we all know. That's what's been done in every other system. Go understand retina, then LGN, then V1, then V2, then V3, and then all of a sudden everything, you know, before Marge Livingstone and Doris Chow, you know, fell apart, and Charlie Gross when you hit IT and V4, like what the heck's going on here? Okay. Similarly in motor areas, you know, for all of us, you most certainly included, that think about skill learning, acquisition, dimensions, sequencing, you know, these types of things. You sort of start off in primary motor, premotor, and then you keep pushing forward to supplementary motor, or even more, you know, dorsolateral prefrontal cortex if we bring decision making into the system, you know, how much is done each place. So I'm sorry, that's a bit long-winded to actually say nothing, but that's sort of what we're struggling with, and very intrigued with trying to take a crack at, but systematically, slowly.
Okay, thanks, Krishna. I think you had like 10 more minutes. You have 10 more minutes left, sort of. There's
I do. Yes, I do.
Okay.
Yep.
So let's see how many we can hit.
Thank you. This is fantastically fun. I certainly don't want to keep anybody, but I'm thrilled to keep chatting.
Yeah, no, a lot of people are staying, so we should keep this going until you have to run. So this is from Chiwei Dong. They said, thank you, amazing talk. Do you think this decoding algorithm would work on languages with more complicated alphabets? If not, what would be a good way to improve the algorithm?
Yeah, so I love this question. By the way, I got this wrong, just as a funny aside. When it became clear that we'd probably be talking with maybe slightly more reporters than we had wanted to, but nevertheless, it's nice to share the science that our, you know, taxpayers pay for. That's the number one question. That's the number one question, and it makes sense why, but I would not have guessed that ahead of time. So here's how I'm processing the question, which is, let's take kanji, okay, or Sanskrit, okay, or Tamil, okay, as Amy Orsborn and her fantastic postdoc wrote in a News and Views article on this paper a couple weeks ago. We think we can do it because we very much view it as decoding movement segments. So if all of a sudden you're making a complicated kanji character, which we have to remember does not correspond to a letter, it corresponds a little bit much more closely to a word, right? Because so for all the detailed pen strokes to create a character, you're getting effectively a word out, okay? So it'd be okay to, for example, take 2 seconds to do that. So in 2 seconds, if you could decode a word, instead of needing 1 second to decode each one of 5 letters to constitute a word, that could actually work much better. The challenge, of course, is the fidelity and precision with which you can decode every little bit. But having been so surprised, honestly, that we could decode rapidly, you know, with even 200 neurons. I mean, if you give me 1,000 neurons, I'll say, yeah, I'm more confident. But with only 200 neurons, where you're always up against noise limits, to be able to decode even English language like that was a surprise to me. So I certainly would not want to bet against being able to do a different language. Okay, here's the problem. Why haven't we already run this? Okay. Well, you know, if you ask me to write kanji, I'm going to be flummoxed. Like, okay, I'm gonna have to like show me the character again, and because I'll process it like a picture, I think, and I'll have to draw each. You know, as a child, I tried to learn Sanskrit and I failed miserably because of the same problem. So we need a participant that is fluent in multiple scripts. That would be the dream. Just like with the speech work, we would love to have a bi or multilingual person, okay? And we internally, just for fun, we refer to this as like our twin experiment, right? In genetics, the holy grail are the twins, right? Because they're genetically identical, but there's variances. So we have a very hard time adding that additional screening factor to what's already a challenging participant recruitment process, because people are quite disabled. It's a great question. We don't see a reason it couldn't work, but we don't know that it would work.
Cool. I guess maybe last question. This is from Xinjiang Song. Thanks for your talk. How dense of an electrode array do you think is appropriate to collect enough useful data?
Yeah, that's a great question. So there's an interesting potential seeming paradox in the talk, okay? And that is, on the one hand, I was saying up front, we need more neurons, more and more neurons, right? I have an infinite appetite for neurons. We all have an infinite appetite. We want more and more neurons. But then you come along and you show with 200, you can handwrite, okay? Maybe you can decode speech. So what's up? Do you only need 200? Should you give up on more like, okay, absolutely not. Absolutely not. Handwriting, cursor movements, simple speech decoding like this is what I would still very much call simple, early. I don't want to quite say trivial maybe, but really not nearly what we can do. So let's instead think about, what if you want to control both arms and both legs? Then how many neurons do you need? Or let's say that you don't want to just move a 2D cursor, but you want to move it in 3D with a rotation and a pitch and a yaw, so 5D. The more complex you make the tasks, is what I'm saying, the more neurons you will need to support a certain level of performance. So how many neurons do you need is very much dependent on what tasks you're considering and what level of performance. Now, add on top of that, now this is engineering reality, but add on top of that, that you will have some attrition of electrodes. Some electrodes 2 years from now won't record as well as now. So it's a great idea to have even more capacity, more recordings, as backup. Hey, that's sort of how our brains are built, right? We lose neurons every day. And, we just have enough oversupply, if you want to think of it that way, that it lasts us most of our lives. And, there's no joke about it, obviously. Towards the end of life, often you really are facing cognitive capacity decline, right? Dementia and so forth. So I would personally say that for the next 5 years, I think that, certainly 1,000 and probably up to 10,000 electrodes should be doable. We could absolutely make use of it. We could absolutely get to higher performance, and that would be awesome. And have we seen this start to happen? Absolutely. I mean, so many of our colleagues are sticking in multiple neuropixels, okay? So for just electrical recordings, people are routinely recording thousands of neurons at a time. And of course, the magic of optical imaging and 2-photon imaging, people are recording from the better part of the brain, right? A million neurons is the current record, okay? And hundreds of thousands of neurons. So while I wouldn't want to fuel an arms race, like the person that has more neurons, wins. That's not the right way to think of it, but we do need more and more neurons. I'm absolutely standing behind that.
Okay, so I think it's almost 15 to very early your time. I don't remember what time it is over there. So
No problem.
We should probably let you know, there's a couple questions and I have a few, but I guess we should let you.
Okay, well, I could, maybe we can let everybody go because I know it's late there. I'm deeply appreciative. Thank you so much. I can stay for another 5 minutes if that would be helpful to some. Yeah.
Okay, then I'll read I guess the last question, which was related to this. Was like, let me check. Maybe, of course, now that we're in a rush, I've missed the question. Oh, so this is from Jiyu Zhang. Thank you. Would it be possible to use electrode arrays to monitor the whole cortex at a single neuron level? So I guess this. What do you see this happening anytime soon?
Yeah, no, not anytime soon. You know, there's been some very interesting papers. But sorry, I don't mean interesting like coded language for crazy. That's not what I mean. I mean, they really are interesting papers of how you could start to approach recording from every neuron. And I think that can be very tough electrically because, you know, at some point you displace a certain amount of volume. So if you really were to stick enough electrodes in to record from everywhere, you displace so much volume, you'd have a problem. But, you know, as nanomaterials and nanofabrication increasingly make things smaller and we're more and more clever and we don't have to record electrically only, like we were saying, it could be optical or chemical and everything. So I think we have a long ways to go, but I have not yet been able to convince myself that realistically we'll ever be able to record from all neurons. Save for, you know, what should absolutely immediately win the Nobel Prize, which is some non-invasive way to record at the single neuron level, and then you could do that brain wide. You know, that would be spectacular. You know, two photon, I think, is probably the closest to that I think that we'll see anytime soon. Yeah.
Okay, thank you, Krishna. There's a lot of thanks also in the chat for the amazing talk and beautiful work. So yeah,
thank you for the kind words.
Yeah, on behalf of everyone, and it was a pleasure having you. I hope to see you around soon. Okay.
Great. Thank you so much, Juan. Thank you, everybody, for extending into your early evenings there. Take care. Bye-bye.
Thank you.